We're kicking off our journey with the Enterobacterales, a group I like to call the gut gang, because they represent the most clinically significant family of gram-negative bacteria we encounter in medicine. This is a diverse family that spans the full spectrum, from harmless, everyday residents of the human gut to some of the most aggressive and invasive pathogens known. What unites this gang is a shared identity. Every member is a gram-negative rod, a facultative anaerobe, and, most importantly, oxidase-negative and capable of fermenting glucose. They also reduce nitrates to nitrites. These simple biochemical traits underpin how we recognize and identify them in the microbiology lab. Differentiation between the Enterobacterales starts simply in the lab with media like McConkey's agar, which quickly separates the lactose fermenters, like Escherichia coli or E. coli, from the non-fermenters, like Salmonella. Xylose lysine deoxycholate agar, or XLD agar, is also valuable because it is a selective and differential medium designed specifically for the isolation and differentiation of Salmonella and Shigella from stool samples. Because culture morphology and gram stain alone cannot differentiate members of this family, biochemical testing, commonly by oxidase, indole, triple sugar iron, urease, and citrate tests, provides the functional fingerprint that allows accurate identification, differentiation, and clinical interpretation. The oxidase test is a rapid and valuable screening tool in microbiology because it enables early differentiation of gram-negative bacteria and guides subsequent identification. Its main value lies in distinguishing Enterobacterales, which are uniformly oxidase-negative, from oxidase-positive gram-negative organisms such as Pseudomonas, Vibrio, Campylobacter, and Neisseria. This early separation prevents unnecessary biochemical testing and helps the laboratory technician select the appropriate diagnostic algorithm. The indole test is a biochemical test used to determine an organism's ability to break down the amino acid tryptophan into indole using the enzyme tryptophanase. The organism is grown in a tryptophan-rich medium, and after incubation, Kovach's reagent, or Ehrlich's reagent, is added to the culture surface. If indole has been produced, it reacts with the reagent to form a red or pink ring at the top of the medium, indicating a positive indole test. If indole is not produced, the reagent remains yellow or colorless, indicating a negative result. The indole test is particularly useful in differentiating members of the Enterobacterales, such as distinguishing Escherichia coli, which is indole positive, from Klebsiella and Enterobacter species, which are indole negative. The triple sugar iron test, or TSI, is a differential biochemical test used mainly for the identification of enteric gram-negative bacilli. It assesses an organism's ability to ferment 3 carbohydrates, glucose, lactose, and sucrose, and to produce gas and hydrogen sulfide. TSI agar is prepared as a slant and butt, allowing evaluation of metabolism under aerobic conditions on the slant and anaerobic conditions in the butt. All organisms capable of fermentation first utilize glucose. If only glucose is fermented, acid production initially turns the entire medium yellow. However, once glucose is depleted, aerobic metabolism of peptones on the slant produces alkaline products, causing the slant to revert to red while the butt remains yellow. This pattern is reported as Ka and indicates glucose fermentation only. If the organism ferments glucose as well as lactose and/or sucrose, acid production continues in both the slant and butt, resulting in a yellow slant and yellow butt, and the pattern is reported as A. Organisms that do not ferment any of the sugars metabolize peptones exclusively, producing alkaline reactions in both regions, giving a red slant and red butt, or a K pattern. Gas production during carbohydrate fermentation is detected by cracks, bubbles, or lifting of the agar, while hydrogen sulfide production appears as a black precipitate due to the reaction of hydrogen sulfide with iron in the medium. The urease test detects bacteria's ability to produce the enzyme urease, which hydrolyzes urea into ammonia and carbon dioxide. Ammonia increases the medium's pH, causing the pH indicator to turn bright pink, indicating a positive result. A negative result shows no color change. The citrate test determines whether a bacterium can use citrate as its sole carbon source. Bacteria that possess citrate permes metabolize citrate and produce alkaline byproducts, causing the indicator to turn blue. If the organism cannot utilize citrate, the medium remains green, indicating a negative result. Now, let us meet our first member of the gut gang, the modal Escherichia coli, E. coli. It is truly versatile because it can be a friend or a deadly foe. E. coli is a normal colon resident but also the single most frequent cause of urinary tract infections, making it a constant clinical concern. Its pathotypes are incredibly diverse, causing everything from traveler's diarrhea to severe hemorrhagic colitis. First up are E. coli strains that cause watery diarrhea. Enteropathogenic E. coli, or EPEC, stands proudly on a pedestal it has built for itself. Using bundle-forming pili and intamin, it attaches tightly to intestinal cells and wipes out the microvilli, creating classic attaching and effacing lesions. The result? Poor absorption and infantile watery diarrhea. Next comes enterotoxigenic E. coli, or ETEC, the vacation-loving troublemaker. Armed with heat-labile and heat-stable enterotoxins, or LT and ST, ETEC doesn't damage cells, it hijacks their signaling. Chloride and water rush out into the gut, causing profuse watery traveler's diarrhea. We also have in the watery diarrhea gang enteroaggregative E. coli, or EAEC, calmly stacking into thick brick-like layers. Using aggregative adherens fimbriae, it forms stubborn biofilms on the intestinal surface and releases the enteroaggregative E. coli stable toxin 1, or EAST1 toxin. This leads to persistent diarrhea, especially in children. Finally, meet the silent infiltrator, diffusely adherent E. coli, or DAEC. Instead of clustering, it diffusely spreads across the intestinal lining via adhesins. The villi become elongated and dysfunctional, leading to watery diarrhea, mainly in young children. We'll shift to strains causing bloody diarrhea. First comes enterohemorrhagic E. coli, or EHEC, ruthless and precise. Armed with Shiga-like toxin, EHEC doesn't invade cells, it kills from a distance. The toxin shuts down protein synthesis, damaging intestinal blood vessels and leading to hemorrhagic colitis. When the toxin reaches the bloodstream, the consequences can be devastating, leading to hemolytic uremic syndrome. Next is the master burglar, enteroinvasive E. coli, or EIEC. Like Shigella, it invades intestinal epithelial cells, escapes the phagosome, and spreads directly from cell to cell. The result is intense inflammation, ulceration, and bloody diarrhea with fever. We also meet in this group the chemist behind the chaos, Shiga toxin-producing E. coli, or STEC. Brewing Shiga toxins, STX1 and STX2, this strain disrupts ribosomal function and poisons host cells. The intestinal lining bleeds, kidneys suffer, and patients risk hemolytic uremic syndrome and thrombotic thrombocytopenic purpura. Finally, we meet adherent invasive E. coli, or AIEC. It doesn't produce classic toxins. Instead, it invades macrophages and persists within them, triggering chronic inflammation. This stealthy persistence is strongly linked to Crohn's disease, making AIEC less dramatic, but no less dangerous. In the lab, we can easily spot E. coli because it's a lactose fermenter, making rose-pink colonies on McConkey agar. On TSI agar, it readily ferments glucose and lactose, turning the entire tube bright yellow from top to bottom. Gas bubbles are also produced, but E. coli never makes hydrogen sulfide. E. coli is indole positive, however, it is both urease and citrate negative. Moving from the versatile E. coli, we encounter Klebsiella pneumoniae, the undisputed non-modal capsule king. Its defining feature is a thick polysaccharide capsule, which gives it a mucoid appearance in culture and makes it highly resistant to ingestion by immune cells. Clinically, Klebsiella is famous for causing severe lobar pneumonia, often resulting in that characteristic thick, bloody current jelly sputum. Beyond the lungs, it is a major cause of urinary tract infections and serious liver abscesses, especially in patients with compromised immunity. Like E. coli, Klebsiella is a strong lactose fermenter. On McConkey agar, it produces large, pink, mucoid colonies due to lactose fermentation in a prominent polysaccharide capsule. On TSI agar, it happily feasts on glucose and lactose, turning both the slant and butt yellow and releasing gas that cracks the agar. Klebsiella is indole negative, yet urease and citrate positive. We now shift our focus from lactose fermenters to a non-fermenter that employs a different strategy, active invasion. Unlike the previously described organisms, which primarily cause disease through toxins or local infections, the modal Salmonella is the true invader. This organism actively invades intestinal epithelial cells and, critically, can survive and multiply within our macrophages. This ability to hide within immune cells allows it to spread systemically throughout the body. We have two main clinical presentations: gastroenteritis from multiple strains and the more severe enteric fever, or typhoid, caused by S. typhi. Salmonella typhi sneaks in with contaminated food or water, quietly slipping past stomach defenses. In the intestine, it invades M cells like a covert agent, then hides within macrophages, turning the body's defenders into its transport vehicles. Riding through the bloodstream, it spreads to the liver, spleen, and bone marrow, setting up secret bases. From there, it launches waves of systemic infection, causing prolonged fever and toxemia. When it returns to the intestine via bile, it triggers intestinal inflammation and ulceration, leaving behind the classic picture of typhoid fever. In the lab, Salmonella is a non-lactose fermenter, producing yellow colonies on McConkey's agar. On XLD agar, Salmonella typhi appears as red colonies with black centers because it produces hydrogen sulfide, which reacts with iron salts in the medium to form a black precipitate in the center of the colonies. This characteristic appearance on XLD agar helps distinguish Salmonella typhi from Shigella, which appears as red colonies without black centers. On TSI agar, it nibbles only glucose, resulting in a red slant over a yellow bud, with no gas but a black precipitate, indicating hydrogen sulfide production. Salmonella typhi is indole, urease, and citrate negative. Not all lactose non-fermenters are systemic invaders. Some are minimalist killers, such as Shigella dysenteriae. The non-modal Shigella is highly virulent with an extremely low infectious dose. You only need to ingest as few as 10 organisms to get sick, which is why it spreads so easily from person to person. Its primary weapon is the potent Shiga toxin, which shuts down protein synthesis in host cells. This results in severe, bloody diarrhea, a condition we call bacillary dysentery. The key difference from Salmonella is that Shigella does not spread systemically, it stays localized to the gut. In the lab, Shigella species shares the non-lactose fermenter status with Salmonella, appearing as small, pale, colorless colonies on McConkey's agar. On XLD agar, it appears as red colonies without black centers. On TSI agar, Shigella ferments glucose, producing a red slant on a yellow bud, but no gas or hydrogen sulfide production. It's also indole, urease, and citrate negative. Next, we'll look at another lactose non-fermenter with a unique motility pattern, Proteus. Proteus, the swarming driller, is known for its unique motility and destructive enzymes. Proteus is instantly recognizable in the lab because it swarms across the agar plate, forming characteristic concentric rings. This is a visual cue you can't miss, but the real clinical threat comes from its urease activity. Proteus breaks down urea into ammonia, which dramatically raises the pH of the urine. This alkaline environment is the perfect breeding ground for struvite stones, also known as staghorn calculi, which can be devastating to the kidneys and are a major contributor to complicated urinary tract infections. Proteus species on McConkey agar produce pale colorless colonies because they are lactose non-fermenters. Unlike on blood agar, swarming is inhibited on McConkey agar due to the presence of bile salts, so Proteus appears as discrete, smooth colonies rather than spreading waves. On TSI agar, Proteus quickly snacks on glucose, only producing a red slant over a yellow bud with black precipitate, denoting hydrogen sulfide production, but no gas is formed. At the indole test, the reaction depends on the species. Proteus mirabilis and vulgaris are indole negative and indole positive, respectively. Then comes the urease test, Proteus's favorite station. It rapidly splits urea, flooding the medium with ammonia. The tube turns hot pink almost immediately, with a strong urease reaction, a signature feature of Proteus. Because citrate utilization varies among Proteus species, the citrate test has limited diagnostic value for their identification. We next introduce Morganella morgani, a gram-negative bacillus that is part of the normal intestinal flora in humans and highly related to the Proteus tribe within the Enterobacterales. Under normal conditions, it behaves as a harmless commensal organism within the gut. However, when host defenses are compromised, such as in hospitalized, elderly, or immunocompromised patients, Morganella can become an opportunistic pathogen, causing infections including urinary tract infections, wound infections, and septicemia. A key virulence-associated biochemical feature of Morganella morgani is its strong urease activity. The organism produces large amounts of urease, which hydrolyzes urea into ammonia and carbon dioxide. The release of ammonia raises the pH of urine, making it alkaline. This alkaline environment promotes the formation of struvite stones, contributing to complicated and recurrent urinary tract infections. From a therapeutic perspective, Morganella morgani presents a clinical challenge due to its intrinsic resistance to many beta-lactam antibiotics. This resistance is largely mediated by inducible AMPC beta-lactamase production, which limits the effectiveness of penicillins and early-generation cephalosporins. As a result, treatment requires careful antibiotic selection guided by susceptibility testing, particularly in the serious healthcare-associated infections. Morganella is a lactose non-fermenter, producing yellow colonies on MacConkey agar. It is indole positive, urease positive, and citrate negative. On TSI, it produces a red slant over a yellow bud, consistent with glucose fermentation only and no lactose or sucrose fermentation. Next up is another hospital threat, Enterobacter. This organism is a major cause of healthcare-associated infections, causing urinary tract infections, pneumonia, and sepsis. The critical point here is its inducible beta-lactamase production. Enterobacter can activate this resistance mechanism when exposed to certain antibiotics, rendering it suddenly resistant to drugs such as third-generation cephalosporins. This inducible resistance makes treatment planning very challenging because what looks susceptible one day can become resistant the next. Just like E. coli and Klebsiella, Enterobacter is a lactose fermenter on MacConkey's agar. On TSI agar, it ferments glucose and lactose, producing an acid slant and acid bud with gas formation and no hydrogen sulfide. Biochemically, it closely resembles Klebsiella but differs in that it does not produce urease. Let's move on to Citrobacter, another opportunistic member of the gut gang. We've seen how Enterobacter uses resistance to survive. The motile Citrobacter is a classic opportunistic pathogen that waits for the host to be weakened, especially in our most vulnerable patients. This organism is notorious for causing severe infections in neonates, specifically meningitis and brain abscesses, which carry a very high mortality rate. While it can cause UTIs in adults, its role in neonatal sepsis and its predilection for the central nervous system in newborns is the high-yield clinical point you need to remember. Citrobacter shows variable lactose fermentation, which can sometimes confuse identification in the lab. On MacConkey agar, Citrobacter species typically form pale pink to colorless colonies, reflecting their variable lactose fermentation. This variability on MacConkey agar helps differentiate Citrobacter from strong lactose fermenters like E. coli and Klebsiella, and from consistently non-lactose fermenters such as Salmonella and Shigella. On TSI agar, Citrobacter ferments glucose, lactose, and sucrose, producing gas and hydrogen sulfide, especially by Citrobacter frundi. In the indole test, reactions are species-dependent. Citrobacter caseri is usually indole positive, whereas Citrobacter frundi is indole negative. Citrobacter is generally urease negative, though weak or delayed positivity may occasionally be observed. Most Citrobacter species are citrate positive. Next, we meet a very colorful character, Serratia, the red pigment rebel. The motile Serratia, classically Serratia marcescens, is a threat to everyone in the hospital, but it has a unique calling card: its striking red pigment, prodigiosin. This red color is most visible when the organism is grown at room temperature. Clinically, Serratia is a significant cause of healthcare-associated infections, often associated with contaminated hospital equipment or solutions. It's frequently involved in respiratory infections and urinary tract infections in hospitalized patients. On MacConkey agar, Serratia is typically a lactose non-fermenter, but that vibrant red color is what makes it stand out. On TSI agar, it ferments glucose but not lactose or sucrose. It does not produce gas or hydrogen sulfide. Serratia is indole negative. The urease test is usually weakly positive, which helps differentiate Serratia from strongly urease-positive organisms such as Proteus. It is citrate positive. Our final gut gang member for today is Yersinia. Yersinia species are gram-negative bacilli that possess effective mechanisms to evade the host immune response. One of their key virulence strategies is the production of Yersinia outer proteins, or YOPs, which are injected directly into host immune cells via a type 3 secretion system. These proteins disrupt cytoskeletal function, inhibit phagocytosis, and suppress intracellular immune signaling pathways. As a result, macrophages and neutrophils are unable to engulf and kill the bacteria, allowing Yersinia to survive and multiply within host tissues. We will focus today on one Yersinia species, Yersinia enterocolytica. Yersinia enterocolytica has a unique ability to grow at low temperatures, including refrigerator temperatures. Unlike most enteric pathogens, it can multiply in refrigerated food, particularly contaminated pork products, milk, and water. This psychotrophic property explains why Yersinia infections can occur even when food has been properly refrigerated, making contaminated food a hidden source of infection. After ingestion, Yersinia enterocolytica targets the terminal ileum, with a particular tropism for Peyer's patches. The organism preferentially adheres to and invades M cells, which normally function to sample luminal antigens for the immune system. Through this route, Yersinia crosses the intestinal epithelium and gains access to underlying lymphoid tissue, where it multiplies and triggers inflammation. The infection commonly leads to mesenteric lymphadenitis, especially involving lymph nodes near the terminal ileum. This inflammatory response produces right lower quadrant abdominal pain, fever, and tenderness, a presentation that closely mimics acute appendicitis, particularly in children and adolescents. This clinical resemblance is a classic diagnostic clue for Yersinia infection. Yersinia enterocolytica is lactose non-fermenting, producing colorless colonies on McConkey agar. On TSI, it typically produces a red slant over a yellow butt, indicating glucose fermentation only, without lactose or sucrose fermentation. It does not produce hydrogen sulfide. It is urease positive and motile at 25 degrees Celsius but non-motile at 37 degrees Celsius. The real challenge in this gut gang is the profound antimicrobial resistance profile. This resistance is evolving rapidly, driven by the spread of enzymes such as extended-spectrum beta-lactamases, or ESBLs, and carbapenemases. These enzymes chew up our best antibiotics, leaving us with fewer and fewer options. This means we can't just guess at treatment. We must use culture-guided therapy and double down on infection prevention and control measures to stop these multi-drug-resistant strains from spreading. We have to stay ahead of this evolutionary arms race. So, what have we learned about the gut gang? They are a bustling cast of gram-negative rods where every test is a clue and every color tells a story. From pink-loving lactose fermenters like E. coli, Enterobacter, and Klebsiella to pale, sneaky non-fermenters like Salmonella, Shigella, Proteus, Morganella, Serratia, and Yersinia, the lab becomes a detective arena. Clinically, the drama escalates. E. coli starts with watery diarrhea but can still shift to bloody diarrhea. Klebsiella hides behind a mucoid capsule. Proteus swarms and makes stones. Salmonella invades and spreads. Shigella strikes with low-dose fury. Citrobacter invades the baby's brain. Serratia flashes red rebellion. And Yersinia enterocolytica chills in the fridge before mimicking appendicitis in kids. Master the panels, follow the colors, and you crack the case, because in microbiology, biochemical tests don't just color tubes, they reveal identities.